This 3-minute video will cover the paper Scalable Continuous Evolution of Genes at Mutation Rates Above Genomic Error Thresholds from the Woldring Lab at the University of California, Irvine, published in 2018. The paper addresses the issue of in vitro directed evolution being not scalable and quite onerous, and in vivo evolution being great but incredibly slow. The authors create an orthogonal error-prone DNA polymerase system which enables continuous directed evolution of a gene of interest without affecting the host DNA polymerases and the host genome. Here, orthogonal means a system of engineered components that interacts with each other without impeding the native functions of the host cells. This is important because they want to have a high mutation rate of their gene of interest without having a high mutation rate of the host genome, which would cause the cells to die with too much error. The engineered DNA polymerase achieved a 100,000-fold faster mutation rate than the genomic DNA polymerases, and they were able to do this without changing the genomic mutation rate. Interesting, in their protein engineering step, they limited the initial engineered DNA polymerase mutational diversity by looking only at the motifs known to affect the transcriptional fidelity before later incorporating mutations to other domains of the DNA polymerase. They then took their orthogonal DNA polymerase and DNA plasmid, a system they call OrthoRep, and conducted 90 independent replicates of yeast transfected to express the malarial dihydrofolate reductase, or DHFRs. They applied selective pressure to each well of cells by gradually increasing a drug concentration of an anti-malarial drug. Out of their 90 independent replicates, 78 of them achieved drug resistance by mutating the DHFR. The rapid in vivo evolution in replicate enabled investigation of the experimental evolutionary trajectories of the DHFRs. The authors then mapped the fitness landscape of a combinatorial library of the 5 most common adaptive mutations. This led them to identify a fitness plateau of various combinations of these adaptive mutations, and also allowed them to identify mutations that were not previously assumed to be beneficial for this protein adopting drug resistance. They also found a unique mutational pathway by removing the possibility for the most common mutation to occur. Ultimately, this research is demonstrating the usefulness of the orthogonal error-prone DNA polymerase and DNA plasmid setup, and its usefulness in both evolutionary campaigns for proteins and investigating evolutionary pathways for drug resistance.